Oh, there we go. It's recording now. So you would just go to this website, go to the software, pick up VMD and NAMD. If you go to their actual page, there's a lot of different options. Typically, I always suggest maybe don't get the latest releases and just go for the one previously. It's usually more stable. And sometimes they may change some random parameter that will kind of break your system, especially if you work with both NAMD and VMD together and they didn't update NAMD in time. It may just cause all sorts of problems. So I usually always suggest maybe one version down and just pick it up for your particular operating system. But once you download that, as Dr. Valfar says, it's freeware. So anybody could just pick it up and just load it onto their particular system. So once you get it, install it, and we'll load it up. So with VMD, as Dr. Valfar said, it's a tool used to design, model, visualize, and analyze biological systems. So proteins, complex protein structures, nucleic acids, RNA sequences. It just allows you to visually see the system. So for today's practice, I just created a really quick polypeptide chain for us to look at. Just to get the basics. So this is the polypeptide. And as you'll see, it's just a very simple 9 amino acid polypeptide chain. You could just easily rotate around it, see it in a visual 3D space. You could tell it to translate if you just need to move it off your screen. You could even zoom all the way in to see particular atoms that you may be interested in. And I am going to change it to make it a little bit easier to see. Oops.
So you're gonna comment on this part, right? This may not be obvious to everybody.
Oh yeah, not yet. This just make it a little bit easier. So now we could actually see how the amino acids are. And say that we want to look at a specific, atom in the chain. We may choose, we just go over the mouse and we'll actually literally pick that atom to look at. And VMD always comes with an interface, a display panel right here, and also a terminal where you can just read that information. So I clicked on this carbonyl, or this carbon atom, and it'll tell you exactly what it is. So it's part of the lysine amino acid. It's the 9th residue of the chain, or I'm sorry, of the polypeptide. And it is, yeah. So you could literally pick any atom and it'll display that information for you. Let's see. And then if you want to go further and you need more information, you can actually label those particular atoms as well. So say you want to label that one right there. And it's a little hidden there, right? So we'll want to try to see if we could see it. If that doesn't work, just moving your screen around, you could literally go to graphics and then labels, and then pick where that lysine was, and then just offset it off so you could read exactly what it is. This could be used for, whenever you're writing your paper or drawing it on your own in like MS Paint or something like that. Looks a little bit more professional too, so let's see. So from here, let's look at more things that we could do with VMD as far as the graphics are concerned. So I think you saw a little bit earlier how we started when we initially loaded the PDB file in. All we got were the simple lines. And some people can probably look at this and they're okay with it. Me, I kind of am a visual learner and I actually like to really see, each individual atoms, where the bonds are connected to it. And I like to be able to visualize and look at it in that aspect. You can easily change that by just going to graphics representation and then adjusting the draw method. They have a variety of options here. I know for me personally, whenever I like to display this information to people, I typically, go to a new cartoon or something like that to actually see what it would probably look like in like a real world system. And then they have a bunch of little different options like that. But some other interesting things that you can do with VMD on this is that you can specifically look at items of your protein itself. So right now, sorry, I have the hiccups. Right now we're looking at the entire protein, so we're looking at it all. But as I said, it's a 9 amino acid chain. And I think my chain is like glycine, alanine, and lysine in that combination. So for instance, maybe you want to see the first 3 residues of that chain itself. All you need to do is tell the program, I want to see the residue of the first one to the third one. And it'll cut it down to those first 3 residues of the chain itself. And then you can further confirm it if you want. As we saw earlier, we just pick it, click on it, and we'll see that there's lysine. It's the 3rd residue. There's the glycine. It's the first residue. So we could show it all, and then we could obviously go back and show the entire thing again. But as I said, I made it into 3 different amino acids in a continuing chain like that. So say maybe I want to see, only the glycines in it. I could just tell it, I want to see just the gly. Oops. Oh, sorry, res name gly. And it'll show you just the glycines that are along that chain. So this is a really small example, but say you're looking at a bigger, longer, more complex protein structure. This may be more useful, especially if it's like huge protein, you just want to see these certain items. Little things like that can help you narrow down the picture so you can get a better view of it. And there is an actual dictionary of all the possible terms you can look at. If you don't know them off the top of your head, like I don't sometimes, you could actually go to the selection tool right here, and then just look at all the possible options that you can use to narrow down your search in this visual field. So it could do almost literally anything you could possibly think of. Let's see, you go down to individual elements as well. You just want to see the carbons. You would just click carbon, apply. Oops. Oh, you have to parse that. That's a little bit more complicated, but yeah, you get the general idea. It's a dictionary of terms that you can use to narrow down your search in this display. And
Chris, can I make a request? I think
sure.
It's something that I also do very often. So would you mind making a demonstration? Sorry to put you on the spot here, but I'll help you out if you don't know how to do this. Fairly certain you can. Just display that entire thing such that all the
I know what you want. Yeah.
Yeah, make all the glycines one color and everything else to be something else.
Yeah. So say you want to just show the glycines in this image. So you'll do res name gly. And so now this one in particular is going to only show those glycines. You can change that color ID to any color you want. And for this instance, we'll just do yellow. Oh, sorry. And we have to set this to the VDW.
I think just doing the.
There it is.
Yeah.
I just had to pick. Oh, I picked the wrong one. You have to, whichever one you change, you actually literally have to click that particular one. So that one I hit name, and I should have done color here into yellow. And then on the all, you can just change that back to type. And then you'll see here, all the yellow ones are your glycines in this picture, like that. You'll probably see later on whenever I showed my little SARS or my COVID example, I wanted to show the exact genes that I'm mutating, and I'm going to do something very similar to this, but I'm going to make it in the new cartoon image that we saw. But yeah, this is a great way to just highlight specific atoms that you want to show in particular in a system like this. But again, all you have to do is just specify exactly what you want to do. So make sure you select the particular item, and they can just change anything in the coloring method, essentially.
Yeah, and to create different representations, all you do is just click on that create rep button, and it just creates different representations. Each representation can have different selections, different attributes, different colors, different representation. So you can literally do anything you want in annotating your molecule.
Yeah, you'll hear me probably say during this workshop how this program is kind of an art as much as it is a scientific endeavor. You're only limited by your imagination, really. They give you so many tools that I'm constantly learning as I go about this every day when I play around with this program. So yeah, I would say if you want to start learning this, just literally throw yourself into it and just start clicking things, scrolling your mouse around, just choosing different options, and you'll slowly get the hang of it and learn all these little tricks and tips along the way, more so than I could ever go through in the next hour and a half. But just to keep pushing forward, as you saw that we loaded this PDB file into this program, and so VMD takes in PDB files. So a PDB file is just like a protein data bank file. All that is just a text file that basically describes the atomic coordinates of your protein, right? So it'll tell you exactly where this carbon is in space, what kind of atomic connectivity it has. Does it connect to a hydrogen? Is it connect to another carbon? It literally tells the program it is exactly at these atomic coordinates, and then it literally builds out the structure for you. So there are a bunch of known protein structures in the world that have been found via, NMR spectroscopy, X-ray. Brad, what's that word? X-ray spectroscopy. And it's all currently housed in PDB. If you quickly just Google PDB, it will go to the Protein Data Bank website. And depending on what you want to study, if there's anything in particular you're interested in, you could just literally search in this protein data bank, and it'll just show you a list of different items that you may be interested in. So, something that's kind of interesting these days is SARS-CoV or COVID-2. So just search in there. They'll send you out all these protein structures that have been associated or found with that protein structure. And you can literally download any of these files. So for the instance of today, I think I will work with something I'm familiar with, which is 7K7P, which is the non-structural protein of SARS-CoV-2. So you'll look up that protein structure. This is the website. It shows you where it is. It shows you the method it was found with. And then you can just literally download that PDB file. So we'll download file, PDB format, save it to your computer. And then you can load it right into your program. So I should probably go file, new molecule, browse for it. It should be in your downloads folder. So here's 7K7P.pdb. Just load it in, and then you have that structure. That's the non-structured protein of SARS-CoV-2. So from here, we've learned how to load in a protein data bank file. We've learned how to manipulate the structure inside the program. Now we're going to move on to more advanced topics that we were discussing earlier, which was NAMD, that other program we had you download. And as Dr. Valfar said, it's basically an add-on program that works in parallel with VMD. So NAMD is more of the calculatory, the side that does most of the calculations for things like bonding energies, things like that. Whereas VMD is just the visualization tool. So what's going to happen is we'll load the file into VMD, but we're going to send our information to NAMD to do the calculations, which will then shoot it right back to VMD to show us the actual visualization of what's happening, right? So to do that, though, we need to basically clean up our PDB file. And so what we need to do is create something called a PSF file, which is a protein structure file. And so what that is it'll basically take the structure, your PDB structure, and it'll create an exact duplicate of it. But it'll also, if there's any like missing atoms or missing bonds, it'll just correct any weird anomalies that may be in that file. Because we download it from this protein data bank, normally you won't, the PSF file usually won't like really correct anything because it's pretty much already been cleaned out. However, if you're like building your own protein, which you can do in VMD, but we just don't have enough time to do it, you may have some irregularities that need correction. This is the part that will basically fix it. But anyways, I'm going on tangent there. So we need to make a PSF file for us to work with NAMD. So how you do that is you load in your PDB file, you go to extensions, analysis, or I'm sorry, modeling, and then go to automatic PSF builder. So this will load up. If you freshly installed VMD, you'll probably have a shotgun of topology files. It's basically a bunch of different files that are of specific parameters to create a, what's the word? Parameters that are more catered towards your end goal, if that makes sense. So there's some topology files that you want to use for like RNA sequencing, some that you want to use for like real world living cells, things like that. There's just a whole bunch of them. I even think I have a collection of them because I was just interested.
Let me add a little bit to that. So in your study, you may want to, for example, study one protein in relation to water. So in that regard, you just want to have all the physical parameters that define the properties of proteins and water. Then you can proceed about your calculations. In other studies, you may want to study proteins interacting with RNA. Then you need to have the topology files for proteins, RNA, possibly water. So each one of these classes of molecules, they have their biophysical properties studied, compiled and tabulated in these topology files. So if you look carefully, you will see things that are classified based on proteins, DNA, RNA, or nucleic acids, based on less well studied would become carbohydrates, and then from there lipids. There is also ongoing research, which really everybody is bringing their focus on. The facilitator of all enzymatic activities is water. So even though it's a dinky little molecule, it defies all of our understanding of biophysical properties in numerous ways. So people focus on studying water. So sometimes people want to study molecular interactions under different parameterization of water, in which case they have different parameters for water, for example. A little bit slightly more than probably you need at this point, but depending on if you're studying carbohydrates, you need to include carbohydrates in your study topology. But in this particular instance, it's just protein. Sorry, Chris, I'm going to be quiet now.
Oh, no, that's perfect. And it's just as Dr. Valvar said, it's really catered towards your particular study. I know whenever I initially started doing this sort of research for SARS, I was reading a couple of articles, and in them they would tell me exactly which topology file they use themselves and freely sent it out, and it's available for you to look at. And so that's basically where I started. I originally stuck with something, generic that VMD provided for me, but then I looked at their particular parameter files and I found that I got a lot more, I felt, more accurate results because it's more catered towards what I wanted to look at. But yeah, it's why, or you simply just have to like Google or just use a scientific article that provides a particular parameter file that may be catered towards your particular research. And that can all be adjusted inside of the PSF. Just add it and just make sure that's the loaded input file. For today's lesson, we're just going to use a generic one that VMD gives us. For right now, we'll just keep it simple and we'll just do I'm feeling lucky. It's just going to use our basic default options and create us a PSF file from that PDB. Oh, so you'll see here that they actually added a couple of atoms from the original PDB file we had, and they just added an additional one, maybe just to, as I said earlier, to get rid of any anomalies that may occur whenever we run through NOMD. So let's go ahead and get rid of that to make it cleaner and easier to look at. So we'll be working with this PSF file. And so what we want to do is look at the energy or calculate the energy of this system, right? So we could do that by going to extensions analysis and then NAMD energy. It'll come out with this box. So we have to make a selection. So we'll look at all of it, of the entire thing. And then let's say we want to look at the energy, and we'll look at all these various energy calculations I could do: van der Waals, electron, non-bonding, conformational energies, dihedrals, yada, yada. Then we just hit run NAMD energy. And then in our terminal, it'll spit out our results. So it looked at all of the bonding energies, the angle energies, dihedrals, the van der Waals, and then gives us our total energy of the system itself. More negative numbers or lower numbers means a more energetically favorable structure. But just like with whenever we're working with the graphical representation and the selection, we could do the exact same thing in NAMD energy as well. Say we only wanted to look at, the first 1 to 50 residues of the structure itself, right? We'll just type that in the selection and hit run NAMD energy, and it'll give us just 1 through 50 of that structure instead of the entire thing. And that's how you could, look at specifically what you want to look at or analyze what you want to look at. You just have to give it direction and tell it what to do, and the program will do it for you. Quick side note, might go off on a tangent, but if you made that auto PSF and you created your own or you use someone else's topology file, you have to make sure that the matching parameter file is the one you chose as well. So you'll probably download both of those, topology file and parameter file, and you just have to load it in. They have to match, or else it will be miscalculated due to it basically moving your atoms or not knowing which atom to start with. Just a side note there. I see a lot of people run into that issue, so want to make sure I get that talked about. So far we've been working with proteins in a vacuum, basically, right? But as we all know, that's not real life. You're typically going to be working with, proteins in an environment that it's in like a solution or something like that. You want to basically model and simulate and get data from a more realistic environment. You can do all that in VMD. So let's go ahead and clear all this out and load back in our original PDB file. So this is that 7K7B file. So we still need to look at energy, right? And when we need to look at energy, we have to create a PSF file. So we're going to recreate it, but instead this time we want to put it in aqueous solution. So it's all built into VMD, where if you go to options, you could add a solvation box to it. So just put it in water, right? We'll do the exact same thing we did. We'll hit, I have to reset, but add a solvation box and then hit I'm feeling lucky. And now we'll create a PSF file for us, but puts it in aqueous solution. And this might take a little bit because it's a big file. Boom. So now it's just in, your H2O here, all around the box. So it just sets up your protein inside of that. And if you have a slower computer like I do, you may want to just hide all of this water. And as we saw before, we could just quickly go to graphics representation, and in selection, all we want to look at is our protein, right? Oh, I'm sorry. We have multiple options here. There we go. So here's our file. I set to protein, but it used to look like that. But we just want to look at our protein, and it makes our system a little bit faster because it will be showing all of the movement of the water molecules itself. So where was I? Okay, yeah. So now that it's in its little aqueous solution, we could rerun our energy calculations and see if there's a difference with the energy. Does water affect the overall energy in any way? And then we see that it's negative 6,000 whatever, 6,000. This is a false because if you notice that we selected all on this, so it's taking into account every single thing in your screen, including all of these water molecules. And all these water molecules, they love each other. They're fine. They want to hang out with one another. So that's why you're going to get this super crazy negative number. But we actually want to see the energy of our protein itself, right? So then we'll
Chris,
yes.
Sorry to interrupt. Did you discuss whether the total energy, whether positive, negative means what?
I have not. I think I just said the lower the value, the more energetically favorable. Okay. But I was probably going to say more towards the end.
Okay, let me just fill in that, because that becomes a critical thing to look at. It's really the foundation of the whole thing. So can I just
go ahead.
A couple minutes and thank you. So the general idea here from the physics point of view is that if a system, any system, doesn't really matter, if it has positive potential energy, that means it has stored energy inside of that system that it is waiting to be released. And when released, it's going to manifest itself in the form of either kinetic energy, means moving, or it's going to be chemical energy, it's going to react with something, or it's unstable system essentially. However, if a system has negative potential energy, which is what we are calculating here, we are calculating potential energy of the system, the more negative it is, the more stable it is. That means you can perturb it and energize it, excite it, meaning you can heat it up, you can irradiate it, you can shine light on it, you can do whatever you put energy into the system. It's going to absorb it and it's going to continue to be stable and not move until you provide up to that negative 6,000 kilocalorie per mole, or whatever the units of energy are. Then at that point, I'm sorry?
Oh, kilocal per mole.
Yep. Kilocalorie per mole. And at that point, once you exceed that, then you start tipping this into the positive direction, and then things begin to move and things become unstable. Therefore, the general hypothesis of the system, of the general area of computational biology, is that we are looking for a state that has the lowest state of energy. Therefore, more negative, the better. The end.
Exactly what he said. So if we scroll up a little bit of where we originally were. So remember when we did the NAMD energy of the protein, but not an aqueous solution, we saw that its total energy value was negative 91. Now that we placed it in an aqueous solution and we're just looking at the protein again, we run NAMD, and then we'll see, there's not much change, negative 91.3765. Interesting. Water didn't really affect it all too much.
Chris, that's not surprising, because at this point you surrounded the water with stuff, the protein with stuff, and then you ask the question of what is the internal energy of the protein? Well, the protein never changed. Nothing about the protein has changed at this point. You haven't minimized it, you haven't perturbed it. So its internal energy remains the same as before. Its energy as a total system, if you want to throw water into the system, then it becomes even more negative, only because of the atoms that are on the surface of the protein, they interact with water, and therefore they are happier now.
Yeah, that was what I was originally thinking, that the water was going to affect it a little bit, but
it is going to affect it if you let it undergo structural rearrangement. So
yes.
Right. So
all right. So now we know how to calculate the energy of our protein itself, now we can move on to molecular dynamic simulations. So molecular dynamic simulations is a function of NAMD itself. So whenever you work with it, you'll need to work with PSF file like we made. But I guess we could talk about a little bit of what molecular dynamic simulations is. It's basically a simulation that analyzes the physical movements of atoms and molecules and things like that, where we allow it to freely move on its own and interact with one another. And basically over time, as they allow it to interact with one another, it's almost like we're allowing it to dynamically evolve, like the system evolves on its own. So, you may have an atom here, an atom here. They get to work around one another and try to find like a favorable spot that they might be more conformationally more suitable for one another, like that. So that's what we're about to do. Let's see. We will first start by going to extensions, simulations, NAMD graphical interface, and I'll bring this up. The first thing we usually do in molecular dynamic simulations is to find we do minimizations, which is trying to find a point of energy minimization in the structure itself. So usually what I'll do is maybe about 10,000 steps of minimization. And I think this computer is going to run it for a while. And so I went ahead and ran it earlier today to show you what's going to happen. But basically, you will go to this page, you'll set up your minimization, and then you'll hit run NAMD. And then once you finish that, you will get another file. Sorry, give me one moment. We're going to load up that 7K7P file. Here it is. Okay. Once you hit that NAMD button, you'll get a bunch of information back. What you'll want to do is load back in your AutoPSF file. So you'll get a autopsf.pdb, and that's the original structure. Like we said, it creates a duplicate of it. Gosh. And then we'll load the PSF file onto it. Right? All you have to do is keep browsing for those two files. And then from here, you'll browse. Oh, crap. And you'll want to add what is called your .dcd file. Right here. Oh, this one. And that's just your updated coordinates from your minimization. Ignore that one for now, but you'll get one DCD file back. I just have this other option that we'll use later on. But once you hit OK and load into this, it's going to load in all of those minimization steps that occurred whenever you perform this simulation. So if you hit load, you'll start to see what happens whenever it minimizes. You can kind of see how the water is being crunched in. And it's loading each single frame of the simulation itself. So I think I did 10,000 steps here, but it may have been 20. Okay, there we go. So 201 frames that occurred. So now you are, if you want to look at it, in graphics, if you want to just look at the protein, you could see that it's changing a little bit during the minimization process itself. But we now want to actually look at the energy and see if it's actually minimized or not, right? So we'll go to NAMD energy like we did. We'll want to look at our protein. We'll want to go for the total energy. And then we'll also, on this one, we'll plot the output. Originally, we didn't select this one because we only had, the one frame of our PSF file. But now we want to see, every single frame along that time slot that we gave it of 10,000 minimization steps. So we'll hit plot output and then hit run NAMD energy. Give it a second to think. And then I'll look at all the energies for each individual step, 192 all the way to 200 frames. and it'll plot the energy for you. So we're gonna look at the black one, which is our total energy. And then we'll see that it energetically minimized. And for minimization, we want to try to see where it kind of plateaus. So there's a little bump here. I think I'll feel more comfortable probably around this time step of 100. You see it kind of like settles in at that negative 21, 50, 55 to 660 range. So I think it's okay to say, around time step 100, it hits its minimum. And with that, we can then continue on and do molecular dynamic simulations, actual molecular dynamic simulations to it, right? So it was I think our original was 10,000 minimization steps, and we wanted to stop at 100 time steps. So we really only need 1,000 minimization to get there. And then you would select molecular dynamic simulations, and then you'll choose how many steps you want to actually perform the simulations on this structure itself. I think for today, I will set it to just 20,000. And before I actually perform this molecular dynamic simulation, I do want to add a little bit extra information for you guys about some of the parameters that you can adjust to your molecular dynamic simulations. So if you hit edit and then ensemble, you can come to this place where you can change kind of your environment. You could set your volume, your temperature, and your pressure to the environment. Typically, you'll want to find an equilibrium for these whenever you do these molecular dynamic simulations. And so, especially in like a live cell, you'll probably want to have a realistic environment. I think that's our goal whenever we do these simulations. So you would typically want to actually set or apply a constant pressure to the system itself, because if it's like a live cell, for instance, like SARS-CoV, it's going to be in an environment where there's atmospheric pressure that's going to be applied to it and things like that. So you usually want to set that to like NPT or something. If you're just going to do it in vacuum, I think NVE's totally fine. You can change the temperature to, whatever temperature you want. But yeah, that's just something to note from there. And also, you can change other simulating parameters to it as well. Especially if you have like a slower computer or older computer or something like that. You change the amount of step frequencies that DCD file I told you about, how many times it takes a picture of each step of your simulation. And you can also change the time step as well. Basically, increasing it will take a little bit less strain on your computer. It doesn't have to constantly, monitor the simulation itself. If you want to look more into it, there's a lot more information about how to make these simulations more better for slower computers and things like that. But for our work today, we're just going to keep it very simple: 1 femtosecond, 50 frequency steps. So yeah, you set your minimization, set your molecular dynamics to 20,000, then you'll hit run NAMD, and then it'll actually run. But to save time, because it'll take my computer quite a while to do it, I will actually load up the file for you to show you exactly what happens.
And if I can fill in the blank there, the reason Chris is actually getting out of the application and getting back on there is VMD sometimes gets confused. When you load new data sets, it kind of has internal memory of what has happened. So sometimes it's better to just start fresh the application, so all its historical information is gone. So if you ever find peculiar errors, just get out of VMD and restart.
Yeah, you know how whenever your internet cuts off and you call the cable company and they tell you just turn off the power and turn it back on? VMD is the exact same way. Whenever I get an error, the first thing I do is just quit and then redo what I was about to do. And by magic, it works somehow. But so we're doing exactly what we did originally. We load up that PDB file, that AutoPSF PDB file. And then we'll load on top of that our PSF file. And then after molecular dynamic simulations, it's going to shoot back another DCD file. This one was, I actually relabeled this earlier. So this was my MDS file that we were going to work on. And so once we load that into it, we'll just let all those frames load in that we captured. Oh, quick tip, especially if you're using a larger complex like this, it's better to just hide all of that water and just look at protein, and you'll see it immediately shot up in frames a lot faster because it's less to actually show you. There we go. Okay. And now we will do the exact same thing. We'll do NAMD energy. We want to look at our protein. We want to look at all of them. We want to plot them all. And this one's a big one, so it's might take a minute. Okay, so now we'll see. We see it dropped, and this was our minimization step. And so once it stopped down at that base point, then we hit the MDS. It shot back up, and then it's performed molecular dynamic simulations. You'll see that, sorry. It constantly was changing and moving and growing and rocking itself the entire time. And it showed each little individual point. And so if you follow this line, the total energy line, and look at your terminal right here, you could see each step and what the energy was as it went. And it kind of looks like maybe the energy is growing, just by eyeball. If I were to continue looking at this, just from, I guess, experience, I'd probably want to increase the number of simulations because it doesn't seem like it plateaus anywhere, in my opinion. And it could possibly continue to grow higher. It may eventually find a spot where it's like maybe this right here will just like flip over, and that's more conformationally, or structurally a better situation for it. And then you can see the energy drop. But another thing I could look at to kind of get a better idea of, hey, have I found a structure that, is more stable or something like that, is you can actually look at how much the structure has changed, if that makes sense. So what that is we look at the backbone RMSD value of it. So if we we'll click that, we'll go to extension and analysis. And I'm just showing you like just the depth of VMD. It has so many options for you to look at and analyze in your own way. You can go to RMSD trajectory tool. And then, oh, nice, it already adds it, but if it hasn't added your file, just hit add all. Then we'll want to look at the backbone, the structure. And then the frame reference, we'll want to start at 0 where we started. And then all you have to do is hit RMSD. Or it did not save it. Okay. All RMSD. Oh, sorry, plot. Duh. You want to plot it. And then I'll show you this. So what this shows you is that you'll see it has an RMSD value of maybe like 2 or something like that. So what it's showing you is that it starts at the original structure at 0. And then as simulations occur, how much has there been structural perturbation, basically, or how much the structure has changed from its original form. And then it calculates out that actual root mean squared value of the change from it. So you can just see that it's still changing, it's still growing. And that, as it increases, that just means it's changing and differing, right? So if we were to eventually see maybe it plateau somewhere, so it just stops out, that just means, oh, there's no longer any structural perturbation. this might, this might be a sign that we've found like the right conformational structure or something like that. So now that I see that's, continually changing and from its previous step in the simulation, it's kind of telling me in my head that, oh, we haven't even, it's still in a it's still evolving, right? It's still wanting to change. There's still more for it to change to. So this is just like a hint to me where I'm like, okay, let's run it more simulations on it. Let's let it keep going, keep evolving, and eventually we may see it actually just plateau off and it hits a formation that might be something more sound, if that makes sense. But yeah, that's just an added tool for you to just look into to kind of create your own hypothesis of, okay, there's more to it here. Maybe we should run more MDS. And honestly, 20,000 simulations, that's low in the grand scheme of things. So that pretty much makes sense. And honestly, that's pretty much it as far as what I want to show at the workshop for today, as far as the tools go. Unless there's more that you'd like to add, Dr. Valfar, I was just going to kind of show some of the research I'm working on right now with SARS-CoV.
Yeah, let me add a couple comments that hopefully may resolve some of the observations that the audience might have made. So if you looked at the energies, as well as the backbone RMSDs, any of those two graphs that you look at, it just seems like it's constructed of two phases. Chris, are you pulling something up? You want to do the energies?
Yeah, I'm pulling up the energy plot for you right now.
Yeah, pull it up. I'll comment on that. Okay, so let's focus on the black line, which is the total energy. Am I correct? Is it the black line? Yeah. So as you can see, there is a monotonous and rapid decay of the energy that gets to approximately negative 1600 kilocalorie per mole. And then there is an immediate and sudden rise of energy that jumps back to approximately negative 1300. And it kind of stays there. So the question is that what is that abrupt increase in energy? And this corresponds to precisely the study, the experimental protocol that we put in place. I think, Chris, you applied 1,000 rounds of minimization followed by 20,000 rounds of molecular dynamic simulation. Am I correct?
Correct.
It's approximately there.
Yep.
So during the molecular and minimization portion, it's purely referred to as molecular mechanics, that you're just looking at the mechanical system and you're seeing how can you move atoms around in space in order to minimize the total energy. During that phase, there is no information related to time. How long does it take to move those atoms? But more importantly, there is no external energy input to the system taken into consideration. Whereas the molecular dynamics simulation, the moment that round begins, temporal information becomes meaningful, meaning that individual atoms, how long does it take for them to move around? The time steps are known. And in addition to that, the system is exposed to a certain temperature. So I think by default, the molecular dynamics simulation is ran at room temperature, approximately 30 degrees or 300 degrees Kelvin. At 300 degrees Kelvin, essentially, there's background radiation that is bombarding the atoms in your system and therefore energizing and exciting your system. And that excitation energy is what you're seeing here. That abrupt increase is due to suddenly you take a system that has no external energy input to the system to a place that is being heated to 300 degrees Kelvin, then you see that jump. And that jump from here may not be completely ground state of the protein at 0 degrees Kelvin, but it is at room temperature, which is the realistic portrayal of what this protein is going to do in room temperature, by the way. So room temperature and physiological temperatures are different. So if you want to go to physiological temperature, then instead of going to 25 degrees Celsius, you kind of need to go slightly higher to approximately, I don't know, 38 degrees Celsius. And that would be the more realistic depiction of what this protein is doing. So this jump that you see here is because suddenly exposing it to background radiation in the room temperature. And it also corresponds, if you look at the backbone RMSD profile, there was a very rapid rise at the very early stages. And from there, it kind of plateaued. It didn't plateau, slowed down. Yeah, so you see that in that first early, I don't know, 25, 150, whatever it is, I bet you it corresponds to the same phase that you see here. And at some point, it's rapid departure because the system is really perturbed. And then when you relax the system, although external energy is kind of added to the system, the backbone is changing, but at a slower pace. So that's the two phases. I know I'm kind of digging a little bit too much into the details, which may not make sense based on the background that we have provided right now, but I'll say it anyways for those of you who may want to go and study this whole system a little bit more. And Chris, maybe you can do that now. So imagine you have a protein, a molecule, a single molecule, it doesn't matter what it is. It's just suspended in midair. And there is no additional energy applied to the system. Well, that particular molecule is going to stay in place. Now you apply energy to this molecule, let's say to your protein. Now, your protein can observe spatial perturbation in two ways. One is just simply maintain a structure, but move in space. This is where you can imagine a ball traveling in space. So if you were to ask the question of how is your atomic positions changing as a function of time, the structure remains the same thing, but because it is traveling through space, either by rotation or translation, the atomic coordinates are changing. Therefore, the backbone RMSD is changing, which is what we are showing here. The second question that you want to ask is that I don't really care about translation and rotation in space. I want to know how much your internal structure is changing. And that's the part that would be meaningful, whether your structure has equilibrated or not. And Chris, if you want to exhibit this one, go back to your backbone RMSD. Leave that one up. That would be a useful thing to compare it to. Just, yeah, that window. Align everything and then compute. There you go. Align and then compute backbone RMSD. And you can see now that by removing translation and rotational contribution to your molecular perturbation, then if you ask the question of internal to the molecule from the internal point of reference, how much is structure changing? Then you can see this, the depicted picture completely changed. There was a very huge rapid change in the early stages, and then kind of equilibration from that point forward. For different studies, different ones apply. So that's where I'm going to leave it at this point. Chris, you can jump into your stuff now.
Basically, I think the takeaway here, honestly, is that just this VMD tool is just so expansive and versatile towards your individual study. What we've shown you here is just the surface of what you can do with this program. There's also additional packages you can add on to VMD itself that others have created for you that could more specify to what you may want to look at. But what we're showing today is just basically a good place where to start. I guess I kind of want to jump into basically kind of where my research is and how what we've learned today I've applied to what I look into in terms of stars. COVID and the simulations with that. So everything we've done so far in terms of molecular dynamic simulations, we've been doing it just by using the GUI provided to us, just clicking, selecting, and things like that. All of this can be done in script, in text, in coding. So what we did here was, we loaded in a PSF PDB file. That's exactly what I'm doing here. I'm just loading those files in. I set my output values and things like that. Set the timestamp or time steps, those parameters that we talked about earlier, right here, and then set temperatures and things like that. And then for scripting, you tell it exactly what we did here, which was minimize 10,000 steps, run this for 500,000 or something like that. Basically, this is fine, especially if you do it on your personal computers and things like that. It just takes a lot of computational resources that just personal use computers just aren't really able to handle on their own for long simulations and things like that. But at USC, we're lucky that we have our HPC clusters, I guess like kind of a supercomputer that I could take advantage of and run much longer scripts and things like that. So for instance, with this for scripting on a couple of these, you can change the temperatures through the scripting. So after I run 500,000 rounds of molecular dynamic simulations, I can quickly tell it change temperature to, or set temperature to, let's say like 1,000 Kelvin or something like that. Take that away. But yeah, then we'll increase temperature, and then we could rerun molecular dynamic simulations for another 500,000. And this way, it'll still spit out the exact same DCD file that we saw from here, but now I was able to make all these changes and tell it to do these things along the way, instead of how we were doing it on here, where we did one step at a time, minimize, then do molecular dynamic simulations. In this way, you could just literally, just set it for this and then go have dinner. And then it'll do all these changes for you and things like that. So a little bit easier, I think, once you kind of get the hang of it. But yeah, I just created these, and then I ran the exact same way, the simulation files, and it shot out a DCD file for me. To make it easier, I then loaded it onto like Excel. So this is the exact same energy plot that we saw earlier with that. So instead of having to run it through VMD, I just took out that data file. I think someone earlier said they were interested in the data aspect of this. It was Non, I think, but yeah, this is what the DCD file actually looks like. It'll tell you the step, the total electronic or van der Waals energy, or total energy, van der Waals, non-bonding, and things like that. And I'll spit it out, and then I just graphed it out. But yeah, that's what I personally looked at on that end. And I think I have, oh yeah, my presentation. So for me, what I was going to look at was the SARS-CoV-2 complex. Did the exact same thing. I loaded in, there's a PDB file called like 70F4, which is what this is. So it's the spike protein and then the ACE protein, the spike being the COVID protein, and then ACE being basically what sits on top of a human cell, where the spike protein from COVID will actually connect to right here. And so, oh yeah, I forgot I labeled it all for everyone. So you can see there. And so if you look at the spike protein, it's actually made up of 3 different chains. They all do the exact same thing, where the chain will connect to the ACE protein of the human cell. So for this instance, the yellow chain is chain B, green was chain C, and white was chain D. But I wanted to look at the chain that's actually connected or interacting with the ACE protein. So the SARS-CoV spike protein that's actually interacting with the human cell itself. So what we did was we wanted to look at that binding energy between the ACE and spike, but then I also, as we saw like last year or even now, there's multiple variants of the spike protein that's happened. And so in VMD, you can actually create those mutations along the way. So if you look at the picture here along the chain, I made all these changes to the mutation. So it's on residue 215 and changed from D to G. I listed all of those in pink right here, so you can see exactly where. And if you guys recall at the start of this, whenever we were changing the graphical details, I showed you how you could specify, I want this residue, I want it to change it to this color, things like that. All done on VMD. So what we wanted to look at, we saw all these changes, but what we wanted to do was look at the interaction of those residues that are actually touching or interacting with the ACE protein itself. And we found those to be 417, 484, and 501. So these three down here still have those mutations, but we're more concerned about that energy interaction between this connection. So take a closer look. It's actually the ones that are literally interacting with the ACE protein itself. So we did, we equilibrated the system like we discussed earlier with minimization. We put it in a salvation box. And then we did NVT and NPT, where we locked in the pressure, and then we also locked in the volume. And we did 500 rounds of MDS for each one of those. And I think you guys already know what it looks like, the molecular dynamic simulations. And then once we ran those simulations, it came back with that DCD file, as we saw, plugged it into NAMD energy, and found, the binding energies that occurred. I went ahead and took the averages of like the last 100 of the simulations and then the overall averages of each one to see what that binding energy was for those three residues between spike and ACE. And so as Dr. Valfar talked about earlier, where you know, the lower the binding energy, the more energy favorable it is. And in terms of, you know, SARS-CoV and the spike protein and ACE protein, it's kind of it kind of shows increased transmissibility because it's just more energetically favorable in that way. So the wild type that we saw, the 70F4, had the overall binding energy of -44, which is, you know, pretty transmissible. But then we saw all these variants that happened. So beta was B1315, which I believe was the South African. Yeah, the South African variant. Gamma, epsilon, and delta. I think this was just delta, not delta plus. But you'll see that the South African variant and B1427, I believe that was the California variant. Yeah, it's the California variant. You'll see that they had they definitely have an increase, but the South African one in particular has almost like double the amount of the wild type. So it can, on a molecular level, it's more transmissible than the wild type itself. So this is kind of interesting to evaluate and see, you know, the differences of these variants of COVID. I think this research was conducted during last summer, so this was pre like Gamma and Delta Plus, but there's a wide variety of things you could do. One thing that we actually talked about to do continued research was doing a mobile and immobile temperature, which what we were thinking was, so whenever we did MDS, we allowed the entire complex, so the we got to delete that. So this was that 70F4, the spike protein structure that we were just looking at. So this huge thing over here were those chains. Actually, I can make it prettier for you guys. Chains, there we go. So as we saw, there were 3 chains to the spike protein and then an ACE protein down here. So whenever we ran those molecular dynamic simulations, we just ran on this entire complex. But wouldn't it be interesting to see what if we took this ACE protein and made it stay still, as in it does not have any simulations done to it, but we instead allowed this spike protein complex just do whatever it wants. So right here, we would just allow only that blue part to just work its way around the ACE protein and things like that. And then what if we stop that and then we made the spike protein stay still the entire time during simulation and allowed the ACE protein to do whatever it wants and move all around the spike protein, if we would see any sort of differences in our numbers and things like that. And I believe that's where we were here, where we were just calculating that out and seeing if there's any changes in the energy levels. I don't know if I completed this research. I fell into a bunch of other work that I had to do, but that was one of the things, further research. But I think the point I'm trying to make here is that there's so many things that you can do in VMD that you could change or tell cells and atoms to do whatever you want. You are kind of like a god in this program where you could control atoms in what you do. And so there's a lot of applications that this can be used. All it requires is a little bit of creativity and a little bit of exploration on the coding part of it. But that's all for me.
Yeah, if I can maybe add a little bit more motivation to what you were doing. Of course, it's not confined to just COVID-19 here. So if one was to look at the life cycle, disease life cycle and mutation cycle of COVID-19. So COVID-19 comes along, we don't know what it is. We can very quickly and cheaply, economically, we can sequence its DNA, RNA, whatever the disease might be. We can get the genetic information very quickly. Now, imagine the transition from the wild type COVID-19 to the next mutation. So that mutation comes along. People on the ground note that there seems to be a different variant of the disease is present. They would isolate it, they would sequence it, and they would know that's a different variant. But in order to answer the question of whether it's more virulent, whether it's more transmissible, they would have to wait for that disease to actually permeate the society. So you have all a good representation of the disease's effect on the population, and then compare and contrast it to the existing group of infected people, and then arrive at the conclusion that therefore South African variant of COVID-19 is far more transmissible than the wild type. And that itself, of course, not only it consumes time, it consumes money, but it consumes also lives. Now imagine at the very first instance that the mutations were observed genetically, so which is very quick thing, within a matter of days you would have that information, and very cheaply, again, within $10,000, $20,000 you would have that information. If this technology to be developed, this molecular interaction calculations, and somebody assessed that whatever we have is a new variant, and this new variant's interaction of the spike protein with ACE protein is potentially twice as much energetically stabilized. And that twice as much does not necessarily mean twice as much transmissible. It could be an exponential growth. So at that point, one could declare this is a new variant that's going to be heavily transmissible, and you do not have to necessarily wait for the disease to actually permeate the society to see what the effects of it is. We can compute how detrimental is that particular disease, and we can intercept and intervene as needed. So these molecular dynamic simulations are going to be the future of studying diseases. My brief preaching.
Yeah, but that's it for me, unless you guys want to go into some really nitty-gritty stuff with VMD. I have what, 20, 30 more minutes that we could do more nitty-gritty stuff. But unless you guys have additional questions, Dr. Fraufer, I'll leave that to you if you want to do anything else. We still have multifactor, like show mutation residue changes, things like that.
Yeah, there's a lot to do at this point.
There's so much.
Yeah, we can stop at this point and see if there are any specific questions from the audience. If not, then obviously this recording is available for you guys to follow, look at it later. Everything that you need is essentially a single computer, free software. You can really do all the studies for $0. The only thing that I'm not sure if it came across is the simulations are very time consuming. So you can run the simulations on your computer and it may take an hour or two or a day, maybe it will return results on a regular desktop computer. However, that 1 day of calculations that you performed is very likely to correspond to only 1 nanosecond of interaction. So the computation time, the interaction window that you observe between between the spike and ACE protein is really in the order of nanoseconds. So in 1 nanosecond worth takes about a day of calculation. So to do a realistic modeling of these, where you want to look at the exposure of spike to ACE protein for a whole second, you have to increase your computation time by 9 orders of magnitude. And that's the primary limitation of this approach, is the computation time. And of course, the solution is to throw it on bigger computers, go to cloud computing, go to massive parallel programming. You can do that, but there are more clever ways that you can reduce the compute requirement in order to achieve the same results much faster. So that's the only thing that didn't come across, that these are not necessarily compute intensive, they are just time intensive. So 1 nanosecond's worth of interaction takes about a day to compute on a regular desktop computer. So we can stop if there are no questions. You guys can review the video. Yeah.
Yeah. Yeah, I have one question in terms of the is there, I understand you have to use all these interactive window. Is there any way you can save the syntax, or is generate the syntax? You can save it and use that to redo or rerun what you've done before with those with these two software?
Right. So everything about the software everything here is reproducible, with the exception of one step. So all of the script that Chris is showing is exactly what you need, but it is also, I think what Chris is showing right now is what this GUI actually generates for you. So the GUI generates it behind the scene for you. If you like, you can further modify it yourself to incorporate some of the more advanced features. And it should be heavily reproducible, with the exception of one spot that at one step of the calculation, there is a certain randomization of velocities, and that randomization is going to change from time to time. And that's the only portion of this entire process that's a little bit on the stochastic end of things. Otherwise, everything is deterministic. Newtonian physics with some initial randomization.
Thank you.
You're welcome. So review the video. Let us know if we can be of any further help. We're always looking forward to establish new collaborations, to transfer the knowledge onto a new system. If there are systems that you're looking at, we'll be more than happy to help out. So if nothing else, I'm going to wait a couple more seconds. Okay. The recording should be made available at some point. I'm not sure how. If you talk to the organizers right now, I recorded the session and put it on the cloud. I am sure they're going to make it available to you. I think the plan was to have it available for the next 2 months or so. And after that, of course, or at any time, we are more than happy to collaborate, to work with you guys. We may have a workshop that spans these topics over a week in the summer. So if you're interested, keep your eyes open. We'll advertise that through Big Data Health Sciences, so you can sign up. It'll be free, it will be remote, and you can do hands-on participation. All right. Well, thank you, everyone. Thank you, Chris, for your presentation. It was very nice. We will see you guys later. Bye.